fun, and we have a packed schedule today, so you don't have to worry about booting up Top Hat or any of those questions because I looked at the schedule and basically it was like, oh, we still have to cover metabolism and the kidneys and fluid balance and the reproductive system, so we're running out of lectures. So I'm going to try to wrap up the immune and digestive system today. Okay, so where did we leave off with the immune system? So with the immune system, we talked about like innate defenses, but now let's talk about B cells. So we talked about T cells the last time, but what B cells do is they have and actually T cells do this as well. So you have your genes, but the thing is that B cells can actually shuffle parts of these genes to make different combinations of nucleic acids and proteins and polypeptides so that they can actually make, in theory, well, it's not completely infinite, but it's a grand number of possible combinations that can result in many different types of protein and amino acid sequences that can lead to different types of surfaces, because antibodies are proteins. So if you start to shuffle the genes that make an antibody, you can actually randomly generate antibodies that have different types of surfaces different types of antigen binding sites. So again, DNA, RNA, and protein. So if you start to do this random generation and shuffling of your DNA, you're going to get random sequence of mRNA, which are used to translate into polypeptides. Now, what does this look like? Well, again, this is like part of the genes that make up an antibody, and antibodies are actually made out of these two what we call heavy chains and two light chains. And I think this is showing a T cell receptor, but it's a similar concept. But what happens is that your cells, your immune cells, like B cells and T cells, before they actually make these type of receptors, they shuffle around these genes that make up these receptors and antibodies. So by having that, they have a random sequence of amino acids and a randomly generated receptor or protein. So that's why you have all these different types of B cells and T cells that recognize all these different types of antigens. Now, do I expect you to know VDJ combination? No, not for this class. That's for immunology. But what I want you to appreciate is that, okay, by doing this shuffling, it's kind of like if you're playing cards, you have a deck. But what happens when you play a game like poker? You draw only 5 cards, and your hand is pretty much random every single time, right? Now, is it completely infinite? No, but you're very unlikely to get the same combination of cards, right? So similar thing with these antibodies and T cell receptors we find in our adaptive immune system. By this shuffling and probability, we get a huge amount of possible combinations, and therefore a huge amount, or I mean, maybe there's a calculation somewhere, but it's not infinite, but it's a huge number of possible antibodies and receptors we can generate from our own cells. Now here we have a bunch of naive B cells, and just like how naive T cells haven't bound their antigen yet, these naive B cells, they have all these randomly generated antibodies, and which are actually their B cell receptors. So B cell receptors are kind of like antibodies, but they're not secreted into the plasma yet. They're kind of like stuck and embedded in the membranes of these B cells. But so these all recognize a specific type of antigen due to that random recombination of genes. But what we have here are all, say you get infected by this pathogen and it's releasing large amounts of this molecule found in its, let's say it's extracellular cell wall or something, like say it's a bacterial protein we're showing here. So which ones are going to recognize it? Well, the ones that are able to bind this antigen are going to be activated by this antigen, but not all of them will be activated because some don't bind this antigen. So the thing is that the chances of, this is why it's important to generate all these combinations and all these different types of B cells and T cells in the hopes that one recognizes some antigen from some pathogen that's in your body. So this B cell right here recognizes antigen, so this B cell will take the first step toward full activation. Not quite completely activated, but it's on its way, so it's no longer naive. So what's going to happen is that this B cell, when it has this antigen bound to its receptor in its membrane, it's going to bring in this antigen, and then it's going to present this antigen on MHC class II receptors. So again, if you remember our whole thing about professional antigen presenting cells, B cells are a type of APC, so they do have MHC class II receptors. So they're going to take the antigen that they recognize on the receptor, bring it in, and put it on a different type of receptor, and that's the MHC class II receptor. Now, what's going to happen now? Well, the thing is that the B cell is it's no longer naive because it bound its antigen using those B cell receptors, which are just embedded or kind of like embedded antibodies. But the thing is that it doesn't want to take the full step yet. It needs to double check its work. So it's not ready, it needs another step. So what's the next step it needs to do? Well, it's going to check it with another type of cell. So it's not going to check it with dendritic cells. So dendritic cells, to refresh and review, they are responsible for traveling to the lymph, or one of the cells that can travel to the lymph nodes, and train naive T cells to recognize and activate them if they recognize the antigen that the dendritic cell scavenged from its surroundings. So if you have this match right here, the naive T cell is no longer naive, now it becomes activated. And one of the things that the naive T cell can become is a T helper cell. So if you have a T helper cell that recognizes the same antigen as this B cell can present that antigen on an MHC class II receptor. And here we have a T helper cell that recognizes the same antigen. So this T cell was already previously trained in the lymph node. So this T helper cell recognizes the same antigen, is like, hey, I saw that same antigen too, and APC showed it to me. So then this B cell is going to be activated by this T helper cell. So now this T helper cell says like, yep, you're not imagining things. I found this, someone else found this antigen. Now you can fully activate and become a plasma cell. So now this B cell is going to take another step, and now it can be a fully fledged antibody secreting cell. So these cells are called plasma cells. So yeah, now the B cell is fully activated. Now it knows that it's bound the antigen itself. It also got double verification from another cell, T helper cell, that yes, this antigen is here. So it's going to now take that same antibody that it generated through that random recombination and shuffling of its genes, and now it's going to start to secrete it into the plasma and the fluid surrounding our cells. So when they have this antibodies, now they can bind to this antigen if it's on some sort of pathogen like this bacteria here. So it's going to bind and target that same thing it recognized when it bound to that antigen on its B cell receptors. So that's going to, and there's many things that can happen from the antibody, but the overall purpose of this is to secrete these proteins called antibodies to neutralize and get rid of this pathogen that's invading the body. So antibodies are proteins, and what do they do? Well, they do many things, but main thing is that they are made against antigens. So there are many possible antigens. In theory, anything that has a molecular surface, it can be an antigen, as long as you can create a protein that's able to bind to that molecular surface. Now, what it can do is what we saw there when we had this coating of this. So what we have with this is all this antibodies are kind of like surrounding and coating this bacteria right here. So that's what we call opsonization. So it's kind of like when you have a, like if you have like a, you're like if you're making like chocolate dipped strawberries and then you're like rolling around in nuts or crumbs or something to some sort of coating. That's kind of like that. Opsonization is like a coating, but it can be, in this case, when we're talking about antibody, it's a coating of antibodies on a pathogen or antigen. Now, the other thing that antibodies can do is recruit complement. So complement are those soluble proteins that can actually attack pathogens by themselves. So again, this is why the adaptive and innate immunity part is like, even though we have those definitions, they won't operate they won't stay in their own lane. They work together, and this is a classic example. So when you have antibodies right here, it can actually cause the complement proteins to target a pathogen, or in this case, this is a infected tumor cell. But what's happening here is this antibody is like the first signal, and then it recruits. It's kind of like saying, hey, complement proteins, come here, we found a pathogen, let's attack whatever pathogen that has this antigen. So this is an example of your adaptive and innate immune systems working together. And it also helps macrophages and phagocytes to actually bind to pathogens as well. Because the thing about the phagocytic cells is that they have little receptors that can grab onto this part of antibodies. So remember, antibodies are kind of like us. They have like they have two arms and two hands that can grab onto antigens. Well, the part that's not grabbing onto it is called the FC region, and actually, don't worry about that for this class. But what I'm getting at here is this other part of the antibody, other cells can grab onto it. So what it can do is, like, here my little macrophage here, and what's this macrophage doing? There's an antibody, and it's able to grab onto this antibody that recognizes this pathogen. So this little antibody kind of acts like a little handhold for this macrophage to eat that pathogen, ingest it, and undergo phagocytosis. Now, antibodies also contribute to something called humoral immunity, and do I want you to know the exact specifics? More just contrast like the two types of immune responses. And what we have here is humoral immunity. So humor is like an old term referring to fluids. So when you secrete these plasma cells, which are fully activated B cells, when they secrete antibodies, they secrete into the plasma. They secrete into the fluid part of our blood. Whereas humoral immunity is often used in contrast to cell-mediated immunity, or sometimes just called cellular immunity. So cell-mediated immunity involves things like those cytotoxic T cells, the NK cells, macrophages. So in those humoral immunity, you have something that's floating around in the blood that's not a cell that's helping with the immune response. So antibodies are an example of that, and complement is an example of that as well. Whereas cell-mediated immunity needs some sort of cell carrying out some sort of immune defense, whether it's adaptive or innate. If it involves a cell, that's the main thing that's causing that or helping in that immune defense, that's cell-mediated immunity. So it's often pretty much anything that doesn't involve antibodies is cell-mediated immunity. I know it's like, but plasma cells produce antibodies, aren't they cells? But yeah, it's like the antibodies, even if you have them by themselves, like say you purify the antibodies and inject them into someone, that can also provide immunity without having the original plasma cells there. So that's why it's like humoral, and when you think humoral immunity, think antibodies and complement, everything else cell mediated. Okay, next so here are the learning goals and key points for here. But basically just contrasting T cells and B cells. So T cells, the main ones I wanted you to know were the cytotoxic T cells, T helper cells, and those T regulatory cells. And contrast B cells. And B cells, I don't want you to go into the weeds too much with them, but know what plasma cells are, and also how B cells are very important for antibody production. Okay, so a little review here. So here we have a pathogen, and all the neutrophils are neutralizing this pathogen. We have all these antigens released by getting rid of this pathogen. Here come the dendritic cells to hoover up these antigens. And what are they going to do? They're going to sample the surroundings, take these antigens, go to lymph nodes, go and find all of these naive T cells where these dendritic cells or any APC can actually train these naive T cells by presenting these antigens. And if these naive T cells recognize the antigen, what are they going to do? Well, remember that they can become things like cytotoxic T cells or T helper cells. But what happens here is like, okay, here we have a naive T cell, and say this naive T cell is lucky. It has a T cell receptor that recognizes the antigen presented by this dendritic cell. So what's one thing this naive T cell can be? It can become a cytotoxic T cell that can go and out into the rest of your body and find cells infected by this pathogen and kill those cells. So that's why they're cytotoxic T cells. But these ones that actually go out here, so when you have this activation of this naive T cell that recognizes antigen, it's going to multiply and when we say proliferate, that's a fancy word of saying, okay, we're just going to multiply these cells and have them increase in number. So these cells are all going to be able to recognize this antigen and kill any cells that are infected and present this this antigen that originally came from a pathogen. So these are called effector T cells because they're carrying out the work. They're actually out there doing the immune function and the cell-mediated immunity to kill any cells that are infected, any sources of this antigen. But the thing is that not all the cells that proliferate from this initial activation, so when you have this activation, some of them don't go off to battle. Some of them hang back. So we call these memory T cells because they're actually not going out to actually carry out the cytotoxic effects. They're just going to hang back. So these effector T cells off to battle, but the thing is that once you clear the pathogen, these effector T cells no longer stay around. They are done with, and they end up dying, and their contents being recycled by your body. Now, memory T cells, what do? Well, they stick around. So even after the pathogen and antigen has long been cleared from your body, these memory T cells hang around, and they still recognize that same antigen. So they're kind of like hanging out in your lymph nodes and hanging out there, just like, okay, threat's over, pathogen's gone, antigen is gone, I'm just going to rest here. But say you get another infection by that same pathogen, and that releases more of that same antigen that was from that original pathogen. So here we have the same antigen coming again, so now it's a second wave and a second infection of this antigen. What's that going to do? It's going to bind to the receptors of these memory cells, and these memory cells are going to wake up. So when they wake up, what do? They proliferate, and they increase the numbers, and they produce all these effector cells again. So why is this very beneficial? Well, you don't have to do the whole thing with training new naive T cells again, because you already have a cell that recognizes an antigen that you encountered previously in your life. By activating a memory T cell, you can go straight to the proliferation and sending out more effector cells. So about 10% of that actually our number is so small fraction hangs around as memory T cells, but the rest end up as effector cells. Yeah, so memory T and B cells, not all of them will become effector cells that deal with the infection. Some of them stick around and hang back, so memory T and B cells. So you can have memory B cells, but I just showed you memory T cells as an example there. So these are like immune records of antigens you previously encountered in your life, and they allow for a faster response because you don't have to do the whole training thing again. And actually that training process is even more involved, but that's for your any immunology classes. So they're kind of like a reserve of T and B cells. So if you do get an infection of that original pathogen and release of that antigen, this memory T and B cells can mount a faster immune response because you don't have to do that whole trying to find a match with some sort of T cell or B cell receptor. Sorry, I know it's like that, but I just want to show you those memory cells, B cells, they wake up. They're very active once they recognize oh, the pathogen is back. Let's go time. So this is why I also want to mention that, because it involves the primary and secondary immune response. So when you have the initial exposure to some sort of pathogen, it takes time. And why does it take time? It's that whole thing with, okay, you gotta deal with that pathogen, you gotta get the neutrophils and hopefully your other cells killing the pathogen. And you also have to train all the T cells and B cells to help with the adaptive immune response to that pathogen. So there is a delay because again, it takes time to process the antigens, present it to T cells and B cells, and then eventually have those B cells become plasma cells that produce antibodies. So all those steps have to occur in this time. But when you have a secondary exposure, what happens? Notice that the secondary immune response, and this is the y-axis here is the concentration of antibody. So you're actually going to produce even more antibodies, and notice that, look at the time between exposure and the peak immune response. Notice that it's a shorter time. And why is that? It's because you have these memory cells, so you no longer have to train all these memory T and B cell you don't have to train new T and B cells again. You can just use your memory T and B cells, and when they proliferate and divide and multiply, especially the plasma B cell or the memory B cells, when they become plasma cells and secrete antibodies again, that's why you have an increased secondary immune response. You don't have to do that training step again. So why is that important? Well, this is the whole thing, the strategy behind vaccination. So vaccination takes advantage of this secondary immune response by actually exposing you to antigens without having the full-blown pathogen that is the source of that antigen. So here we have like a more of the traditional type of vaccine where you take an antigen and then you vaccinate someone in it in the hopes that they generate memory T and B cells that recognize that antigen from that pathogen. So this is like, or now I should actually add mRNA vaccines, because the whole purpose of this is to train your memory B and T cells to recognize that antigen before you're actually infected with that pathogen that has that antigen. So this is the whole purpose of that, yeah. So here we said the naive T cells, effector T cells, and some of the memory T cells hang around with that. So what's the whole purpose? So again, here's our first infection and here's our second infection if you just have like something like COVID, right? So say you get your first infection, what's going to happen? It'll take time for your immune system to generate the T cells and B cells you need to fight that infection. The second infection, hopefully, or something like traditional, like with the flu, what happens is that you generate a higher secondary immune response because you already have memory and B and T cells with that. So the whole purpose of vaccination is not to get you infected the first time, but to cause that primary immune response and train those memory B and T cells around this time right here. So when you have those memory T and B cells, what happens when you're actually infected with this? So when you're actually infected by this and you have this vaccine that trained all these memory T and B cells, you're going to produce even more antibodies, and it's going to take you less time to produce those peak antibodies because you already have those memory T and B cells in advance. So this is where I like to make an analogy between like a vaccination and a study guide. Yeah, you can take the test without a study guide and you can probably pass, but the thing is, like, would you rather have a study with a study guide that helps you to prepare, or would you rather take the test cold without having to prepare at all, with no preparation? So it is possible to clear that test either way, but which is going to help you with your response and recall? Probably the one where it helps your memory even more, right? So that's what vaccinations are. They're kind of like study guides for your immune system, so that when you actually get that pathogen, your immune system can react faster because it's already been trained. It's already has it committed to memory, so it's able to deal with the pathogen faster than if it was doing it by itself. So this is and this is topical because you might hear people saying like, I just want to get this over it. I just want a natural infection and just get this over with. Well, remember, if you have your first infection, it takes time to train your cells and immunity to recognize that pathogen. So which would you rather with your first infection? Would you rather have this immune response that has fewer antibodies and takes a longer time? Or would you rather be vaccinated, and if you do have a first infection, instead of having a primary immune response, you skip straight to the secondary immune response, which has more immunity and is faster to respond. So that's the whole strategy. It's not just about it's not just about let's inject this for giggles right there. It's trying to help your body adapt to this and mount a stronger response and faster response than if you just got exposed naturally. But again, you can still mount an immune response. It just takes longer if you're unvaccinated. Okay. So memory T and B cells only considered part of the secondary immune response since their non-memory counterparts were part of the primary immune response. Yep, that's correct. They are vital for the secondary immune response because memory T and B cells, they are not actively, if they were active, they would be considered effector T cells. But since they're hanging back in the lymph nodes, they are memory T and B cells. So they're important for the secondary immune response. Okay, so that's the whole thing I wanted to talk to you about, and this is why it's very important. Again, this is if you have not vaccinated. I mean, you can try to do it the primary immune response, but that's why it's like that's the benefits of vaccination. It's so that you can mount a stronger and faster immune response versus trying to do it cold without any sort of exposure to the antigens. And memory T and B cells are essential to this, and this is why sometimes you need boosters, because even if you get infected or if you have like an initial vaccination, Sometimes you don't produce enough memory T or B cells, or sometimes you actually need multiple exposures to generate enough memory T and B cells so that you actually get an effective secondary immune response. So that's why boosters are sometimes needed. Sometimes it doesn't take the first time, and that's why sometimes they might do a blood draw to see like if you actually create antibodies, because sometimes you might be vaccinated but you didn't take in your immune system. So they actually need to look at the level of antibodies to see whether your immune system was actually stimulated by that vaccine.